Two opposite pathways are regulated in such a way when one is activated, another is kept in inhibited state. Yes, what I said, two opposite pathways are regulated in such a way. Suppose this is liver. This is liver. We found that in liver, yes, two opposite pathways. Yes, glycogen to glucose, glycogenolysis. The rate limiting enzyme is glycogen phosphorylase. Glucose to glycogen, this is glycogen synthase. These are two opposite pathways. These should be regulated. Yes, glucose to glycogen, glycogen to glucose, these are two opposite pathways. These must be regulated in such a way when blood glucose, yes, one is activated, another is should be kept in inhibited state. Let me start. This is glucagon, epinephrine, norepinephrine. They increase cyclic AMP. Glucagon, epinephrine, norepinephrine increase cyclic AMP. This increase cyclic AMP, yes, activate protein kinase A. Protein kinase A. Yes, and we found that this protein kinase A, kinase means transfer of phosphate group. Kinase transfer a phosphate group. Protein kinase will transfer phosphate group to protein. Protein kinase will transfer phosphate group to protein. So we found protein kinase A transfer a phosphate group. Transfer a phosphate group. All enzymes are protein. So glycogen phosphorylase is protein, glycogen synthase is protein, and you all know few enzymes are activated by phosphorylation and few are inhibited by phosphorylation. So we found that glycogen phosphorylase is activated by phosphorylation and it is inhibited by phosphorylation. So we found whenever glycogen phosphorylase is activated, this glycogen synthase is inhibited. What is the reason? Because we know that these are two opposite pathway. These must be regulated. When this is activated, this must be kept in inhibited state. Suppose a person goes into fasting state. A person goes into fasting state, then glycogen would be broken down to glucose to maintain blood glucose. But suppose this enzyme is activated and this is not inhibited. This is not inhibited. Suppose this is not inhibited, this is also active. Then what would happen? Suppose in liver cell glycogen is broken down to glucose. This glucose will accumulate in liver cell. This will diffuse out of the liver cell. But glycogen synthase is not aware. Yes, I say that glycogen synthase is an enzyme which has instruction. Whenever you see extra glucose, you will convert extra glucose into glycogen. Yes. So this glycogen synthase has instruction. Whenever you see extra glucose, you will convert into glycogen. Suppose glycogen phosphorylase is activated and glycosynthase is not inhibited, then what will happen? This glycogen phosphorylase convert glycogen to glucose. Yes, this glucose will be converted back to glycogen by glycogen synthase. This will not come out. It is breaking, it is making, it is breaking, it is making, and blood will get Babaji ka Tullu. Yes, yes, Kapil Sharma show. Yes, that's why nature, what nature said, okay, whenever we will activate this, we will inhibit this. Okay, so glucagon increase cyclic AMP, activate protein kinase A, attach phosphate to both of enzyme. This is activated, this is inhibited. Clear? Insulin decrease cyclic AMP, insulin decrease cyclic AMP, glucagon increase cyclic AMP, attach phosphate, and you can make mnemonic. You can make mnemonic. Yes, I told you insulin insult phosphate. Yes, I told you insulin insult phosphate. Okay, glucagon attach phosphate with glue. Glucagon attach phosphate with glue. So wherever you will find glucagon will attach phosphate with the help of glue. Okay, so yes, so glucagon, any enzyme activated by glucagon is active in phosphorylated state. So you can say what glucagon do. Glucagon attach phosphate with glue and activate. Attach phosphate with glue, activate. Okay, any enzyme which is activated by glucagon is active in phosphorylated state. Okay, yes, clear, got the point. So this glycogen phosphorylase is activated by glucagon, so these are active in phosphorylated state. Got the point? Now you can easily tell the answer of this question. Yes, this enzyme A in decreased cyclic AMP, A is insulin. Yes, B activates increased cyclic AMP, that is glucagon. So B glucagon activates cyclic AMP, increase cyclic AMP, and insulin decrease cyclic AMP. Clear?